Jaakkonen and I work as a senior researcher at the Finnish Food Authority. I present a joint research project with the University of Helsinki. External veterinarian advisors were also involved from the Finnish broiler industry and animal health EDD. We investigated population genetics of antibiotic-resistant Campylobacter jejuni in broilers, especially the possibility of vertical transmission from parental to production flocks. The story begins in 2014, when we started to see peaks in resistance to tetracycline and quinolones, in this case ciprofloxacin and nalidixic acid. To give you some background, in Finland, both the prevalence of C. jejuni in broilers and their antibiotic resistance have long been below the EU average. In 2014 to 19, we had 6.5% of slaughtered broiler batches positive for the termotolerant Campylobacter, mainly jejuni. Before 2013, less than 4% of the isolates were resistant to quinolones or tetracycline. In certain years after 2013, however, we saw annual proportions of 10 to 25% resistant to quinolones or tetracycline. What was interesting is that production flocks had not been treated with antibiotics since 2010. Antibiotics had been used, however, to treat 3 to 20% of parental flocks annually, with the highest proportion in 2014. Mainly penicillin, but also tetracycline was used. Quinolones were not used. Also in 2013, the import practices and origin countries of grandparental flocks changed. With this background, we hypothesized that vertical transmission was one possible explanation for the spread of antibiotic-resistant C. jejuni, although this was an unlikely transmission route based on the literature. To find out, we sequenced whole genomes of 333 C. jejuni isolates. The isolates had been collected from Zika at slaughter in the National Control Program for Production Flocks and volunteer in-house control for breeder hens in 2014 to 19. One temporally representative isolate per flock was sequenced, covering annually 34 to 74% of Campylobacter positive production flocks and all breeder hen flocks. The genomes were subjected to MLSD, whole genome MLSD, and genotypic antibiotic resistance determination of the genome assembly and quality control. We had also metadata linking production and parental flocks. We found 15 clonal complexes and 95 sequence types. Isolates of certain sequence types were always resistant to quinolones, especially within the clonal complexes 206, 21, and 658. Also, tetracycline resistance was more prevalent in certain genotypes than others, especially within the clonal complexes 206 and 658. Within the maximum of 11 allelic differences, we found 13 clusters that comprised both broiler and breeder isolates, of which 2 clusters with resistant genotype. The 2 resistant clusters involved altogether 19 isolates and 15 farms. Almost all of the quinoline-resistant breeder isolates were associated with these 2 resistant clusters, whereas the majority of tetracycline-resistant breeder isolates didn't belong to any clusters. Conversely, the majority of broiler isolates with quinolone or tetracycline resistance belong to either these clusters or broiler-only clusters. Antibiotic-resistant broiler and breeder isolates were not, however, epidemiologically linked, which contradicts vertical transmission. So, our results rather suggest horizontal transmission from a common environmental source. The farms of each resistant cluster located within the distance of 60 kilometers. To conclude, This study provides new information on the genotypes, resistance profiles and population genetics of Campylobacter jejuni from Finnish broilers and breeder hens. Thank you for your attention.